yeah, so thank you. Hi, so I'm Frank. Um, as I'm a PhD student in computational biology here at CMU and in the University of Pittsburgh. And I've been following this series of the seminar series, and it's actually quite interesting to be now on the other side. So I'm quite happy about that. Okay, so today I named this talk Programming Genetic Circuits that Operate as Neural Networks. And uh, always when I present these talks among my colleagues and among my lab mates, it's been like received. So the concepts are received with a little bit of, I'll say, healthy skepticism. So if you don't mind, I will spend some time trying to contextualize our work into the synthetic biology field. And by the end of the talk, I'll try to convince you all that this is actually an interesting topic to work on and is very thought-provoking. And also, I'll talk a little bit more about applications just by the end. So I took this picture, some fantastic schematics from the biocontrol feedback systems book, which really categorizes all the processes that happen right inside the cells into these 6 categories. And I would like to propose here a subcluster of just 3. So we will try to understand how a cell processes worked through a sensing module, through an actuation module, and through a computation module. And the whole all the talk that I'm going to present today is going to be solely focused on the computation module. And we will approach computation from trying to solve a very particular problem, which is going to be a classification task. And so in particular, the way that I like to see this is that when you have a set of biomarkers, maybe surface receptors, maybe small molecules, maybe proteins, and some other biomolecule, how can a cell tell apart other cells? Or how can a cell could discriminate or differentiate between different cell types? So the way in which I like to formulate this problem is trying to think what an observer cell would be looking, how will it consider this classification task? And this is the kind of visualization that I really would like to introduce right now. This is what the cell, potentially the classification task that the cell will be trying to solve. So we could approach this problem from 2 perspectives, 2 big paradigms of computation. It's either digital or Boolean logic or analog computation. So today, just to be a little bit more specific, we're going to work on analog computation, right? And I think that this plot already hinted a little bit what do we did on this work. So long story short, we worked on classifiers. We built some classifiers. And I think that this previous information, that this system level approach to cell engineering that I have just shown you, it's really helpful, right? Because it will help everybody to understand the rationale behind our designs. So there is this ongoing challenge on building classifiers, and it's related to the dimensionality of the data. And I would like to exemplify that by looking at hematopoiesis. So let's say that we would like to differentiate between, or build a classifier, right, that differentiates between megakaryocytes and maybe monocytes or erythrocytes. So what we could do a priori is do a sort of differential gene expression analysis, right, to know which genes are upregulated or downregulated in megakaryocytes that are not present in other cell types, and we could potentially build a classifier using those. And the dimensionality of the data in this sort of classification task would be, I'll say, in between 10 and 15 genes, but I might be wrong. But the problem, it's more well exemplified when we are trying to differentiate between different cell types that are within the same clustering here. For example, this one's in purple. So cells that share very similar gene expression profiles, but we still would want to differentiate between those, right? And the dimensionality of the data in this case of examples could scale up or add up to maybe 100 genes. That will give a very unique fingerprint or gene expression profile that will make this problem relatively linearly separable. So I have this other example in which it illustrates the trade-off. If we use our current technology, right, that's able to process just a limited amount of inputs, we will need to embrace the fact that our inputs will have a nonlinear relationships between them, right? And here at the right, I'll put just an example on cancer cell discrimination. When we have cancer in red, healthy in blue, if we would like to differentiate, right, between those two cells, the relationship between both is inherently nonlinear. So today, I'll try to argue that genetic circuits that work as neural networks that we have just called biomolecular neural networks or BNNs, but I will refer to them as neural networks just for the sake of simplicity. They're actually quite good at solving these problems. So with the current approaches that we have, to the extent of my knowledge, what we usually do is we mine biomarkers, right? And we find, or we're trying to find these gene expression profiles that fit into what our network or our circuit could do. But if we worked with neural networks, we could potentially adapt our circuit to the data and not the other way around, right? Which I think is the current approach. If we use a single neuron, for example, we could potentially create a linear classifier. If we use one or more neurons, just borrowing some terms from machine learning, we could potentially create some more nonlinear classifier. Now, before going on, I would like to give a little bit of a disclaimer that it's always occurs when we talk about this topic in the laboratory. And I just picked this picture from a very interesting review on control theory in synthetic biology. So our neural networks are not artificial neural networks. They are not biologically informed. They are actually genetic circuits that mimic how neural networks work. So if you want to see it more visually, we are working on the left sort of challenge and not the right one. So if it wasn't clear from the explanations before, I hope that this visualization will make it a little bit more clear. So let's just say some definitions before I want to give you some background information about this, but we got to state some formalities before. So this is the kind of schematics that we will use to talk about a single neuron, or also called perceptron, right? Where the output of this neuron is essentially the weighted sum of certain inputs that passes through a function that's called an activation function. And those activation functions could be the rectified linear unit or ReLU activation function. It could be the soft plus, which has a more smooth transition, or the sigma activation functions. I just picked some examples to make that a little bit more clear. And so it happens that the idea of building neural networks using proteins or using biomolecules as computational elements is actually not, it's actually quite old, but really enough, it's been like very underexplored topics. And Dr. Bray, for example, made this analogy between the sigmoid response on phosphorylation and dephosphorylation processes that are known to exhibit this sort of sigmoid. He made the analogy with activation functions. Arguably, that's the first mention of a biological activation function. But this is whole theoretical work based on observations. In 2003, the last figure of this quite interesting paper that worked on the logic of transcription in the lag operon also suggested that the type of regulation that's done at this transcriptional level also resembles, at least structurally, a neural network. And the weights here are associated with the binding affinity between the RNA polymerase and its binding sites. 1 year later, we got this very interesting, also theoretical paper, where the first neural network using DNA molecules is proposed. And we needed to wait about 7 years until we got our first in vitro implementation in this very, very beautiful work from 2011. I might be missing some papers here in this chronology, but I think these are the ones that have really driven and initiated this field of, some people call it unconventional computing. So just in the interest of time, I will be skipping some of the state of the art, but I'm happy to come back to these sort of implementations after the talk. Actually, this second implementation using proteases have been presented in this seminar, I think some time ago, which is also very beautiful work. All right, so we could try to abstract ourselves and see which kind of circuitry, which kind of chemical reactions have been used for creating neural networks. There are about 32 to 33 maybe papers that have been published since 1995 to date that talk about this topic. These are two, I think, very interesting reviews on the topic, but that might be biased, right? Because one of those are from our laboratory. But we observed that we could abstract ourselves to 4 different chemical networks that implement or could be used to implement, sorry, neural networks. One of those is molecular sequestration, in which we have 2 molecules Y and Z that are able to bind to form a complex C. We have the covalent modification cycle, which I think the most prominent example is the phosphorylation dephosphorylation cycle, right? Done by a kinase and by a phosphatase. We have the enzymatic reactions, right? Pretty similar to molecular sequestration, but just considering that the enzyme is able to recycle itself. Actually, the work of proteases fits into this model. And a sort of relatively new proposal, which is based on competitive binding. So in this work that I'm going to present to you in this talk, we focus solely on molecular sequestration reaction. And in this chronology and considering the other work that has been done on the literature, what we did here is basically we tried to complete the picture. We tried to interface concepts from machine learning to synthetic biology. So in this work, we introduced concepts such as formally the perceptron. We introduced activation functions mathematically. We came up with ideas to do negative weights and showed that they could be done experimentally. And we worked on the concept of decision boundaries as well, because we are trying to solve a classification task. So something that I also want to really highlight about this work is that it had been heavily informed by mathematical models. You know, the usual ones that we solve, that we use mass action genetics to model with, and which we solve. And that led us to not only very interesting properties of molecular sequestration, but also very useful properties. And one of those, which we're going to cover throughout this work, is what we're calling non-steady state conditions or out-of-equilibrium conditions. So in few words, this is how our work is fitting into what has been done right now in the literature. Perfect. So now just going to talk about some experiments. So to begin studying the molecular sequestration reaction, you know, using the formalities of machine learning, so we designed a toy model using DNA hybridization reaction. We have, well, this system consists right into DNA strands that have a complementary binding site, fDNA that has a fluorophore, and qDNA that has a quencher. They are able to hybridize, because the quencher, right, comes in close proximity to the fluorophore, then the complex is not, does not fluoresce. Just for the sake of simplicity and to, you know, introduce a little bit of notation, I'm naming fDNA Y, I'm naming qDNA Z, and I'm naming the complex C. This is essentially a binding and a binding reaction, right? When we solve for the steady state and under the assumption of very fast sequestration, which is very feasible, even real in this kind of cases due to the complementarity of the strands, we get to this expression. So the steady state of our fDNA could be approximated as an operation, as the maximum operation between 0 and the subtraction between the initial concentrations of fDNA and the initial concentrations of qDNA. So if we take these expressions and we map them, we have inputs here and output here. This resembles a ReLU activation function, and I hope you agree with me with that, or at least a function that exhibits a threshold in here. This is when assuming high sequestration. If we do relax our assumption, we could potentially get this sort of curve in which we get a little bit of a smoother transition when we increase the dissociation constant or decrease the sequestration rates, which are equivalent. Let's say now that we think of a titration experiment. We consider different inputs or different initial concentrations of DNA in their horizontal axis and different concentrations, increasing concentrations of the quencher DNA on the vertical axis, right? We could come up with these heat maps And this sharp transition in color, I know that the resolution here might not be that high, but this transition in color here is what we call a decision boundary. And just to relate these two plots, if we trace a horizontal line here in red in our heat map, we will be able to recover this sort of activation function visualization. This is just to introduce all of you with the types of plots, visualizations, and formalities that we're going to use throughout the work. Now, with extreme mathematical cautiousness, let me reinterpret the equations that we have before in the following format. So we will have that the output is going to be the maximum between 0 at some weight that multiplies x1 minus some weight that multiplies x2. Here, W1 and W2 are 1. I missed the unit here. I'm sorry. It's 1 over hour. And x1 and x2 are just going to be the initial concentrations of the DNA strands that I showed you before. And let's keep that in mind for the next slide. So this is all numerical simulations. It's a pretty interesting example to study. When we replicate this in the laboratory, this is the results that we found. So using our DNA hybridization model, we were capable of recovering this sort of activation function that I truly think that resembles a rectified linear unit. And we could also recover this heat map in which I think the most interesting property is that it exhibits, right, this decision boundary. I'll acknowledge again the resolution of our experiments, but I think that's a pretty convincing evidence that we are, in fact, right, looking at this sharp transition. So this interpretation, the mathematical interpretation that I showed you before, is quite versatile. And it led us to design different patterns within the input space. So this is a chemical reaction, just an abstraction of what I showed you before. This is the kind of schematics that we're going to use from now on. X1 and X2, remember, are the initial concentrations of our DNA strands. We have just assigned +1 and -1 to the weights. We will come back to that later, and we have our output here. So if we introduce a biased species that's constant on our reaction, here the biochemical implementation might be a little bit easier. So we introduce, for example, the qDNA in our mixture constant in our grid, and we just vary between different concentrations of fDNA, we will be able to sort of flip our decision boundary that was initially at this sort of 45 angle degree, and now like it's flipped. In gray here, you'll see numerical simulations. Here in blue, you'll see our experimental data. And I feel that this is a very good agreement between our results, which is very interesting. I'll highlight again that we started with the numerical simulations and then we obtained the experimental data, right? And our experiments were heavily informed by these sort of models. So it was very interesting to see that data actually fit in reality. We could do the same, but just inverting a little bit the logic. So now the fDNA is our constant in our reaction, and we have X1 and X2. We will have titrated X1 and X2, and we will obtain these kind of patterns that, again, we were able to replicate that, what we saw on the numerical simulations in our experimental data. Perfect. So far, I think that by now we have a pretty good understanding of the dynamic, well, not the dynamics, but the behavior of the molecular sequestration reaction at steady state. So this is interesting. We have incorporated here a lot of formalities, but this was known, right? The early DNA hybridization neural networks that I showed you before actually have a similar logic. So we ask this question. Do our linear classifiers could operate outside of the steady state assumption? And to do that, we used a relatively more complex model, yet still a toy model in a cell-free system. Maybe this is going to be a little more intuitive and a little bit more interpretable for all of you, because in this case, X1 and X2 are actual plasmid copy numbers, right? They will be encoding some gene that could be transcribed and then translated into a protein. We're focusing here on a protein-protein sort of sequestration reaction. Again, we're calling this Y and Z just for simplicity, and we let them bind and unbind, forming the complex C. So we did our chemical reaction sort of abstraction one more time. Here we have X1 and X2. Remember plasmid copy numbers. They will be producing at certain rates W1 and W2, the species Y and Z, which are proteins, right? When we solve, this time not for a steady state, but actually the dynamics for Y over time, we were able to recover the same expression, right? The same activation function. This is we think the ReLU activation function. It's just a scale over time. And this makes a lot of sense, right? Because we have a cell-free system that in which the species do not decay, do not degrade, they just accumulate, right, over time. And that's kind of like modeled with this parameter. The interesting part here is that we were able to recover the rectified linear units. And there are some very, very interesting properties that we could just come up only by looking at this expression. So the first one is that we have the weights of our network, of our perceptron or single neuron in this case, are the production rates. And this is a very useful observation that you'll see in the next slide. Why? And something that I just escaped on purpose on the previous example of DNA hybridization is the sign here, which is a result of molecular sequestration reaction. And this is super interesting because this is a way in which we can generate subtraction, like consider this negative sign of our networks. And we should remember, right, that, of course, it would be physically impossible that our concentrations or our production rates are negative, right? So molecular sequestration have been very handy in order to generate this sort of negative weights, which is right way closer to what's done normally in computer science. Perfect. So here I'm showing another map, input-output map, similar that I've shown you before. And as you'll see, yes, we are obtaining this sort of ReLU-like activation functions that are scaled due to time, as predicted here. Just showing you some heat maps that I think that look pretty cool. We have our heat map at 1 hour, 3 hours, and 5 hours. And again, this is just a matter of scaling, right? The interesting part is that since the beginning of the reaction, we do see a decision boundary here, right? Remember this sharp transition between virtually zero output and some output above the threshold. And here you can see it. So to do this experimentally, we use the sigma and anti-sigma factor sequestration reaction. So we had two plasmids. One plasmid will be encoding our sigma factor. The other plasmid will be encoding the anti-sigma factor. We worked this on a cell-free system in the TXTL system from Arbor Sciences. After transcription and translation, right, of both, we will obtain the sigma factor and the anti-sigma factor that will sequester each other and just form a complex. So we use a reporter gene in this case to track, right, or monitor the expression, or at least approximate, right, the expression of the sigma factor, which is just DGGFP, with a promoter that's responsive, right, to that particular sigma factor. Here you can see our results for the sigma W and anti-sigma W. Here you can see how does the heat map looks at 3 hours, how does the heat map then looks at 5 hours. Due to space constraints, I couldn't show the whole time series data that's available in our supplementary material, and I'm happy to show you afterwards in case it's needed. Something that's important to mention here, so I am showing you 3 and 5 and not 1 and 6 or 2 and 6. So for these sort of experiments, we used a reaction volume of 2 microliters. Our plate reader wasn't able to pick signals before 1.5 hours. So 3 hours was the most reliable signal, the most consistent that we could come up with. And after around 5.5 hours, our cell-free reaction, the resources in our cell-free reaction get depleted. And so it looks a sort of constant, which is not a steady state, right? It's just like resource depletion. So let's say our working range is from 3 hours to 5 hours. But within those times, we are able to pick up this sort of behavior, right? That were predicted with the approximations before. And I'll say my favorite part is this one. when we, again, this input-output mapping, right, we were able to recover the ReLU-like activation function. This is fantastic because we see this with sigma W and with sigma 28. A little bit less resolution, but here you can see it. My bad, this should have been T equals 3 hours, because the same logic that I presented you before, just another system, right? But this is pretty cool, right? Having observed also the activation function in another system. So why did I make or try to highlight the fact that our weights are interpreted as production rates, or the other way around, production rates are interpreted as weights, is the fact that we now are able to, in a more straightforward way, adjust the decision boundary, right? These are just numerical simulations. For example, we could increase the weight that's associated with x1 or decrease the weight that's associated to x2, and that will give us a higher, right, decision boundary. In the same way, we could potentially decrease the weight of x1 or increase the one in x2, and that would reduce a little bit the decision boundary. So to approach this, we took a very simple approach. We actually just diluted our plasmids for the experiments. And here you can see we diluted the one that's associated with x1. And that also led us to reduce the slope of the decision boundary. We did the same, but diluted the one in x2, and we were able to increase the slope of the decision boundary in the same way that it has been predicted by the models. Again, important to highlight here is that we observe this behavior not in a single time, but actually over, right, certain period of time. Again, because of this out-of-equilibrium property that we found mathematically before. So we could potentially explore the input space. Let me just move this here. Yeah. Again, by adding this constant species to our reaction, that will let us to flip the decision boundary or invert, right, that part that has color, that's called the classification region. Here, for example, we had a constant concentration of plasmid that was expressed in the sigma factor, and we just had two different plasmids that were encoding for the anti-sigma factor, right, which you can see here in the x-axis and in the y-axis. And we will able, right, to sort of flip like this, the decision boundary. Again, very, very good agreement between what we predicted numerically, what we saw experimentally, and once more, we kind of like the out-of-equilibrium property holds, right? Also for this kind of implementations. Happens also for the sigma 28. In this case, we had the anti-sigma factor as a constant, expression at a constant plasmid value in our reaction, and then we titrated, right, over like different values of our sigma factors here, and we were able here to sort of invert the classification region. Same properties hold again. Perfect. So the last thing that we did in this work was do a proof of concept. Our proof of concept was done in mammalian cells, in HEK cells in particular. But we kept our sort of low dimensional model here. We tried to challenge a little bit our model, and it worked surprisingly well. So that's very cool. The way in which we did this modeling was basically considering degradation rates, different degradation rates for each species, right? Due to inherent protein degradation, or maybe due to cell division or dilution. We also considered that our plasmids were diluting over time, because we assume as an experimental setting a transient transfection. You will see how that looks experimentally. We worked the math. We came up with this expression. Once again, very interestingly, we recovered, right, the ReLU-like activation function. And it was just scaled by some, factor that depends on the degradation of the species that we were looking at, Y in this case. This is how it looks, the input-output mapping over time. And here I just pick one to show you the heat map, how it looks when you consider, right, that the plasmids are able to dilute on time, and you consider also degradation on the system. Before showing you the the experimental data, I thought that it would be interesting to provide some insights into the approach that we did to characterize this. So we didn't want to do these titration experiments again, especially in mammalian cells. So came up with this idea, which I think is pretty cool. It is inspired by this paper, the polytransfection paper from 2019. So it's just a very little change in the lipofectamine transient transfection protocol. Basically, you have two plasmids and you combine both plasmids, and then you add the reagents that are needed for doing this lipofectamine transfection, you'll get a sort of correlation. If each one of these plasmids is expressing a fluorescent reporter, you will get a correlation, right? Here shown like in different colors. However, if you create those complex, right? But each plasmid is mixed with the lipofectamine reagent, and then both are transfected, then you get a sort of diversity, right? And we actually harness this diversity to characterize our circuits. So basically what we did is we replicate this with our own circuit. You'll see the design in a little while. And then we bind this system and we recover the expression of the protein that we were interested in, and that led us to recover the same sort of heat maps, right, that you were looking before. There are some statistics that we did here just to check if the BNN that we use is the correct one, if we have like a statistically significant, amount of cells, reliable or we could trust of their measurements or their average measurements at least. So as for the circuit design, this is what we did. I'm sorry, I just took the picture from the paper, but I'm going to explain it. I'm going to try to explain it. So we have two plasmids, right? Let's focus on the plasmid on the left. Is have a promoter, a constitutive promoter that will be expressing tag BFP. So that tag BFP would be used to infer the inputs from before. So it will be one of our accesses. Yeah, so what we did here is we added an RNA stabilizer, we added a target sequence, and we added an IRS sequence that will let us to co-express tag BFP with M neon green. This is, we'll let this transcribe, and when this translates, we will get neon green. Of course, we'll get also tag BFP, but here the logic is focused on neon green. Perfect, and we measure in neon green, of course. In our other plasmid, we'll have the same promoter, but we will have a protein called CSY4, which is an endoribonuclease. We have an IRS sequence that enables us to co-express with IRFP. Once again, that one IRFP would let us to infer the inputs. This is going to be another axis of our plot. So when the CSY4 is transcribed and translated, it recognizes this signal. Here we put this signal, sorry, this sequence, and we put here a little square, and that forms a complex. It sequesters each other, right? Yep, that's basically what we did. We use CSY4, and these are the results. This is how it looks after the binning operation that I told you before. Hope you agree with me that we are looking here at a linear classifier. Here there's a linear decision boundary, and here I'm showing you the activation function, which is a little noisy, but exhibits a ReLU-like pattern. If you don't believe that this is a ReLU-like pattern or are skeptical, at least it shows a threshold, and that's virtually enough to generate a classifier. We also tried with the Cas-E. Here I'm showing you another result, maybe a little bit more noisy, but still interesting to see that it exhibits a sort of ReLU-like input-output map. So, and just to finish, we also worked on weight tuning, right? Based on the previous expressions and following the same logic or a similar logic that we did for the TXTL system. So we did a small experiment. We had neon green co-expressed with tag BFP, and we just added these URF sequences that will make the translation of neon green a little slower than the IRS and the tag BFP. And we wanted to compare, right, how's the expression of neon green using the first one as our control. These are the results that we got. So arguably what's being expressed after the IRS sequence is independently expressed. So here we took the ratio between neon green and tag BFP, just assuming that tag BFP is a constant, then you see that this histogram shift or this distribution shift to the left, meaning that M neon green is decreasing, right, this expression. And this is due to these repetitions of the URFs that we are using. So we pick up the one that has like the highest effect, the 5 URFs. Here you will see, here you will see the reference of the linear classifier that we have. And this is how it looks when we add the 5 URFs, right? Because we are diminishing the expression of the CSY4. If you recall the expression that we used before, this is essentially as if we were increasing the, decreasing the weight that's associated with the X2 variable, which in this case was the CSY4. Yeah, and just to finish, I'll just want to give some insights into, I'll say, some thought-provoking applications of this system. What we do right now is forward engineering, right? We could potentially, what we want to do in the future is to build nonlinear classifiers, right? Which are a little bit more complex than the ones that I showed you before. We have shown that this could be done using the same reactions that I showed, molecular sequestration in particular, and which is a matter of time, to get this experimentally. And another idea that I really like, but I do think that's still very exploratory and it relies a lot of data. It's, I think, some sort of holy grail like in synthetic biology, right? When we have a system that we want to replicate and when we do this input-output sort of mapping, we wanted, right? If we wanted to replicate these endogenous systems using synthetic circuits, we would like essentially to replicate its input-output mapping. There is this theorem in neural networks called the universal approximation theorem that says, right, that that relatively large neural network in width and in depth, we could potentially approximate any sort of function. And that's something that we are like quite interested in exploring by using this sort of neural networks, trying to, do synthetic versions of systems. There are a lot of open questions, of course. You probably have some right now, but we are in the process also trying to solve them, both theoretically and experimentally. So I just want to give some acknowledgement to all the people that work in the lab, all our collaborators. I think I'll need to thank Carnegie Mellon, the Computational Biology Department, and Cold Spring Harbor Laboratory, particularly the Synthetic Biology course that runs every year. Fun fact is that many of the experiments, actually 2/3 of this paper, were done in 2 weeks. And here you have a couple of our authors comparing their sizes with the amount of plates that we use to make all of these experiments. Perfect. So this is everything that I wanted to share. I think I went a little bit over the time, so sorry about that, but I'm happy to take any questions. And again, thanks for your attention.